So let's start, guys. Good afternoon. Good afternoon. So wonderful to see so many faces here in front of you at Herschel Grammar on our second talk on our biology masterclass, and hopefully also people from around 30 schools all over the country. Even we have in Jersey one school, but also in Scotland schools joined our masterclass. So hopefully the streaming and the team is going out there okay. And what I thought I just quickly before my lovely colleague takes over remind you that we put up resources for you. And as I told you, we want this interactive, and we want that you go away and go further than the lecture. So this Padlet is sort of our central resource, which we established. So you can see that, let me see if my camera it is on actually, that so we put up the links how to follow us, the YouTube, the team, and we also have a web page where the program and a couple of other information is there. But then I put up this Science Daily. So the idea with this is that this is a web-based science news possibility where they put very short articles highlighting new discoveries week by week. So what I advise you that you become a scientist if you are following science. And to follow science is to read papers, and I mentioned Nature, Science, but then also it is good to have news articles to follow, shorter pieces, and then maybe if you are excited, they always cite the source and you can go back to the source. So do this Science Daily. You can search this also by topic. So whatever you find interesting here, just see that what is on Science Daily in the last couple of years on that topic. So this one is an important sort of a journal club type of activity. Then I put up a couple of talks, and the purpose of this is that we need to recognize important scientists on a field. Here I talked about cell cycle and cancer, and Paul Nurse and Robert Weinbergs are the giants in this field. So you can learn how they discover things, and who went to this resource because it was up after my lecture. Anybody looked into this? Hands up for this. You need to get active. Yes, so like this one is 5 minutes. Paul Nurse is telling about what he thinks is important for a researcher, and it's not a lot of time to waste from your day. 5 minutes. So go to those things. This is a long little clip, but this is that he likes to think hard and summarizes what is the essence of life. So if you want to be a biologist, you want to make these big questions. What is the essence of life? And and listen to that talk. So then now I established that feedbacks to our talks and questions can come, that you can put comments on this corner. So even during the talk, I found that the team chat box not always working. So online, who is listening, if the team chat box doesn't work, and it's more difficult for us to follow because we have so many gadgets around, that put your comments up, and we can always come back to these questions in the next masterclass. So if we don't manage to find those questions today, then we will come back to that either by writing answer to your questions or by answering before the next lecture. And then I am always asking the next speaker to give me their PowerPoint and their link. So Chris gave me his PowerPoint, Dr. Ryder, and which is here. So you put your comments here, and he gave us two resources. One of them is his blog following the book he recently published on the genetics. So this is more or less, I wanted just as a reminder that this is not a cinema experience, but we want to be an active discussion and working together with you. And I bring now up the PowerPoint. And before I fully give the microphone to Chris, I just want to tell a couple of words so Chris worked with me the something like 16 years before he retired, because he was already there when I came 20 years ago. So I know him very well. He's a retired lecturer and gave wonderful lectures, and still teaching on my course of the cancer as a emeritus member of our team. And he is also interested in many things, and that's why after retiring he didn't stop thinking about science, and he went and wrote a book which he might be talking about. And with this one, I give the microphone to Chris. And yes.
Well, good afternoon, everybody. It's nice to see so many people here with an interest in biology and, in particular, genetics. So I want to focus on genetics. Sort of everybody is aware that a basic property of living things is their ability to pass on their characteristics to the next generation. And genetics is the science of explaining how this inheritance of characteristics occurs. And I'd like to hope that this afternoon I can persuade you that genetics, although it was a relatively latecomer in its development as a bioscience, it's now so powerful that it impacts our everyday life, and I shall talk about some of the instances of that. So I wrote my book, as Latsi says. Unfortunately, it's only out as a hardback, so perhaps your school libraries might consider a copy, but it is quite expensive. It's supposed to be coming out as an ebook. But you also might like to read my blog, What's in Our Genes, which you can search for at drcryder.com, on which I post stories that interest me about recent genetic findings. So, in my book, I decided that the way to explain this to a non-scientific audience was to treat the subject as a piece of history. So, top right here, does anyone in the room know who that is? So this is Gregor Mendel. Oh, sorry, I beg your pardon. You were about to say, of course you were. So thank you. So he was a monk in Austria, and he was really the first guy to get the science of genetics started. Although inheritance is such a basic property, no one had been able to figure anything out. In fact, a contemporary of Mendel's was Charles Darwin, one of the greatest thinkers in biology. And inheritance and genetics was really central to his theory of evolution, but he got nowhere in understanding how genetics worked. And what Mendel performed was some very well-designed experiments for which he did a very thorough mathematical analysis of his results. And he came up with a surprising finding that contradicted the existing beliefs, that when mating occurs, the properties of the parents are not diluted out, but they survive intact in indivisible units. And it's that concept which we now refer to as a gene, a unit of inheritance. How about the guy bottom right? He's probably a little less well known. He was the American geneticist Thomas Hunt Morgan. And he was a person who was able to show that genes exist as an array on our chromosomes, with each specific gene having a particular site on the set of chromosomes in each of our cells. Still had no idea what a gene was or how it worked. So, following on from their work, here are some basic milestones in terms of genetics. So it was about the time of the Second World War that it was established that the genetic material actually is this compound deoxyribonucleic acid, DNA. That is the function of DNA within living systems. It is physically what genes are. In the next decade, by the 1950s, it was worked out how genes basically work. And this is a statement here. Maybe you come across it as one gene, one protein. This is a slightly fancier version, one gene, one polypeptide. But fundamentally, what this understanding now tells us is that each gene dictates the structure of one protein, and therefore each protein has its own gene. This version is slightly fancier because it recognizes that some proteins, an example we may have come across is hemoglobin, have more than one different polypeptide in their structure. Does that ring any bells? Therefore, one gene for each different subunit. So 2 genes to make hemoglobin. Now, there are gaps of roughly a decade or more between all of these major milestones, but don't think for a minute nothing was happening. What was happening behind it is that to enable the next discovery, great experimental advances were taking place in genetics. So in the mid-60s, we had got to the stage where people had figured out how DNA encodes proteins. Basically, each 3 units, a triplet of bases in the DNA sequence, dictates which amino acid should be in the next position of the polypeptide chain. So now we know how they work. And it was also shown that the code by which it works is universal. With very few exceptions across all living organisms, the genetic code is exactly the same. And that's actually very fundamental to some of the engineering advances genetic engineering advances that have been made. Because if you take a mammalian gene and put it in a bacteria, the bacterial system still reads it in exactly the same way, following the same universal code, and it will produce the human protein. In the late 70s, DNA sequencing became a really practical proposition. There had been earlier bits of sequencing of DNA, But the guy that really kicked it off is a remarkable scientist called Fred Sanger. And he is remarkable because he's one of the few people to win not one, but 2 Nobel Prizes. And the second of those prizes came from his ability to sequence DNA by a really efficient method. And his triumph was that he took this bacteriophage. It has this code name Phi-X174. A bacteriophage is a virus that infects bacterial cells. So it's actually very easy to grow in the lab. You grow up a load of bacteria in a culture, infect them with this virus, and you get a lot of bacteriophage and therefore a lot of bacteriophage DNA. And he sequenced the whole thing, the whole genome, the entire genetic blueprint of this virus. Now, in terms of what we can do nowadays, this is quite simple because, like all viruses, this bacteriophage has very few genes, only 11 in its single-stranded DNA loop that it has. And that loop is about 5,500 bases long. So a fairly simple genome, but he figured out the whole thing. And at the turn of the century, the new millennium, the then Prime Minister Tony Blair and the then US President Bill Clinton announced that the human genome had been totally sequenced. Like most political statements, it wasn't totally true, but it was pretty much true. And this represents an enormous step forward, which in turn needed considerable technical advances in genetic experimentation, because now we have 25,000 genes, over 2,000 more than Phi-X174. But there's even more DNA because spacing out these genes are very large regulatory elements of the DNA, which don't actually encode for proteins, so they're not genes as such, but they regulate how those genes will be expressed. So we have 3 million bases, and because it's double-stranded, we call them base pairs. So there's twice as much DNA again to sequence. And by now we have effectively a complete sequence of our own genomes. So rather than go into details of what I've just covered in a rather quick flip through, all of the techniques which underline the advances I've just spoken about mean that genetics is extraordinarily powerful. And it enables us to do things which even a few decades ago would be beyond imagination. So instead of randomly breeding by traditional methods, crops, livestock, garden plants, whatever, these have been bred for centuries, but very much on a look and see basis of what happens by mating different individuals. Now we can target genetic changes, and this is the whole topic of genetically modified foods. It's a complete topic in itself, which is coming up later in this series. There's also another talk in this series on gene therapy. We know that certain diseases are genetic diseases. They are inherited diseases caused by faulty genes. So rather than try and treat the outcomes of these diseases, why not get stuck in and correct the fault in the genes? So again, gene therapy is a topic which you'll come to later in the series. So I want to concentrate on these other issues. And the first topic I'll turn to is this question of making human proteins to treat diseases. And a particular class of proteins which are useful in medicine are antibodies. After all, antibodies are proteins. But I'm going to take the examples of insulin, the blood clotting protein factor VIII, before I get round to talking about antibodies. I'm also then going to talk about applications outside medicine, and DNA fingerprinting in forensic science is based on the advances of genetics. And in this era, it's impossible to consider genetics and biology without contemplating viral diseases and the measures that we can now take against them. So, making human proteins and antibodies to treat disease. So the use of human proteins, or the use of proteins, I should say, to treat disease has a long history. And the disease where this first came into medicine was type 1 diabetes. So as a reminder, we have two forms of diabetes: a childhood form, in which the patient loses the ability to manufacture their own insulin. Insulin is a protein controlling glucose metabolism and the whole energy balance of the body. And this condition develops in something like 1 in 165 children. So I don't know how many pupils you have in your school, but the chances are there's quite a handful of them who would suffer from type 1 diabetes. Now, this is different from the mature onset type 2 diabetes, which is associated with obesity. Type 2 diabetics do make insulin, it's just their body doesn't respond to it. So giving them more insulin is not going to help them. But the type 1 diabetics, the childhood diabetics, require insulin, otherwise they would die a very distressing and prolonged and miserable death from their effectively starving to death, regardless of how much food they're eating. That is until the 1920s, when two Canadian medical researchers, Banting and Best, showed that what is lacking in type 1 diabetes is this protein hormone insulin, and it's produced by a special set of cells in the pancreas, the beta cells. So although this talk is about genetics, it's important to be very clear. Type 1 diabetes is not a genetic disease, it's a cellular disease. If you lose these beta cells, you don't make insulin, okay? So insulin, and here's the structure, is a fairly simple protein. It has two chains, one of 21 amino acids and the other of 30 amino acids, colored in blue and green, respectively. And as protein chains go, these are very short. So it's a small protein. And following Banting and Best's discovery of insulin, it was easily possible to purify insulin and supply it to diabetics to maintain their health. And the source of this insulin was pig pancreases collected as a sort of waste material almost from slaughterhouses. So purified pig insulin was used to treat the human disease. And that worked pretty well for several decades. But there are issues with it. The problem is that as far as the human immune system goes, any pig insulin that's injected is a foreign protein. The problem is not so bad because in this short protein, there's only a few amino acid differences between the human and the pig. So most diabetics, their system would not react by producing antibodies against the pig insulin. But in some instances, it did. So far, we have no genetics involved in this particular story. But now we do. Because the way to make human insulin, genuine 100% human insulin, is to not purify the protein, but isolate the genes which encode human insulin, transfect those genes into a bacterial culture. As I referred to earlier, the bacteria obligingly then make the human protein, we can isolate this from bacterial cultures. And since the late 70s, the manufacture of human, and we now call this recombinant insulin because we're recombining the bacterial genome with the human gene. It's a human gene in a bacteria. This now is the preferred insulin, and all diabetics these days would go straight on to the human insulin, and therefore there are no immunological complications of that. Okay, so here is another disease which is treatable by supplying a protein which is missing, and this one is a genetic disease. And what it concerns is blood clotting. So I don't know to what extent you cover blood clotting in your syllabus. The blood clotting pathway is absolutely essential for our well-being, and it's quite complicated. There are a number of proteins circulating in the blood all the time, and these are referred to as factor, and then the different ones are numbered with a Roman number system. So there are people who have a mutation in the relevant genes, and these suffer from the disease of haemophilia, in which there's uncontrolled bleeding. So you might think that the problem for these people is that if they suffer some external injury, a cut or something, which in most of us the blood would clot after a period of time, seal itself up, and blood loss is controlled. The real problem is not excessive blood loss through external injury, but internal injury. Because as we engage in physical activity during the day, we might bump or knock our limbs, for example. Most of us would develop a bit of bruising, maybe, if it's a bit of a severe knock. And after a while, that bruising disappears, no problems. But because that bruising is the result of internal bleeding, if we now have an individual with haemophilia who cannot control their bleeding, then that bruising will be excessive. And so what these people suffer from is progressively acquired swelling and distortion in the joints as they suffer internal injuries. So haemophilia A is the commonest of the haemophilias. There are different haemophilias because different blood clotting proteins are affected. In haemophilia A, it's factor VIII. The gene for factor VIII lies on the X chromosome. So we males only have one copy of it. If we're unlucky enough, we will be 1 in 5,000 men whose gene for factor VIII is a mutation, and therefore we will not be able to make this. Women and girls with their 2X chromosomes, the chances of acquiring a mutation in both of those genes is vanishingly remote. So it's one of these male X-linked diseases. This is a genetic disease. So the answer would be to give men and boys with haemophilia A, as they will be, the human protein. Now, this is the structure of factor VIII, and I hope you can see at a glance, it's a very large protein with several hundred amino acids folded in this complex structure. So the prospect of using an animal factor VIII is impossible. Any repeat administration of a foreign factor VIII would be recognized by the immune system. An antibody response would develop, and then that protein would be rapidly neutralized and useless. So only human factor VIII will do, so that we don't have those immunological complications. And because factor VIII is a blood protein, a potential source of it, which was used for many years, is to purify it from outdated blood donations. And again, that will work quite well, except for a major problem. And the major problem is that the purification methods which were applied to this were rather crude. And the preparations often contain contaminants, and the worst contaminants are infectious viruses. So there are a number of hemophiliacs who suffered either viral hepatitis or HIV infection because they were being contaminated with the very agent that was being used to treat their original disease. And deaths and serious illness have resulted, and there's still an ongoing public inquiry in the UK as to whether the medical authorities actually should have done more about a problem they were aware of, or they were caught unawares and it was all a terrible accident. But either way, a lot of suffering has been caused inadvertently by this contamination. So again, since the mid-80s, it's been possible to identify and isolate the gene for factor VIII, to transfer that into cultures of animal cells. The animal cells then produce the human factor VIII. That's purified. There's no possible contamination from infectious viruses, and we now have a product that's entirely safe to give. So there are a number of other examples of therapeutic proteins that are used in the treatment of disease, and the story is essentially the same. Either safety from not having to cross the species barrier, or safety from being free from contaminating viruses. These are avoided by using recombinant technology and manufacturing these things in the lab, where all the processes of production can be much more stringently controlled. So I'm going to stop here for questions. Any questions? I'll take questions from the room first, shall I? Yes.
So this is slightly hypothetical question as well. So with both examples you gave, they were more about treatment of genetic diseases.
Yes.
How exactly would you foresee, for example, with genome sequencing, how would you see that in a more preventive?
So the question is that the instances I gave about treating the outcomes of the disease, which is spot on, that's exactly what it is. In terms of future gene therapies of attacking the genes, which would only work in the case of the haemophilia, because that is a genetic disease, then I think there are prospects that future gene therapy processes. But I'm going to skip going into that anymore. You can ask the person speaking about gene therapy if they don't already cover it. Of course, the gene therapy approach would not help type 1 diabetics because it's not a genetic disease. They've lost the cells. Unless you could think of ways of engineering other cells to take their place as the insulin producers of the body. Maybe that's going to be possible, but not done yet. Yes?
On the note of, like, as you said, type 1 diabetes,
Yes.
why does that occur?
It's, our belief is that it's an autoimmune attack on those cells. In other words, some kind of childhood infection perhaps fires up the immune system, and the attack on that basically not very harmful virus somehow gets subverted into destroying the beta cells of the pancreas. Okay? Are there any online questions? No? Okay. So, oh yes, one more in the room. Yes. Yes, and if it hadn't been specially processed, the human immune system would have wiped it out in 24 hours because the barriers of xenotransplantation, to give it its proper name, transferring across the species barrier, formidable. But if you look into that story, those pig hearts have been engineered, and they've had certain extra genes put in to protect them once they're in the human system. But they've also had certain pig genes knocked out, so as to render them more human-like. It's quite a complicated story. I think I'll duck out of going into the details, but certainly that approach would be completely impossible without the genetic understanding of what needs to be done to make a pig heart acceptable. So this is the first example of xenotransplantation. Whether that's going to become a more routine way of providing hearts for transplantation, or whether there were still barriers to it that we haven't overcome, that remains to be seen. But of course, there is the big issue that heart transplantation surgery is now straightforward and routine. There just aren't enough human hearts to go around. Okay?
There is one. So could cystic fibrosis be treated using genetic therapy?
So the answer to that is that experimentally that's been done in animals successfully. So the question is, can cystic fibrosis be treated by gene therapy? So again, I perhaps could leave that one over to the speaker on gene therapy. But I'd like to make the observation that it's quite difficult. In principle, gene therapy sounds really straightforward. But the problem is, as with our beta cells of the pancreas, that when you start thinking about the role of individual cells within the body, getting your therapy to the right place is actually quite difficult. And the problem with gene therapy of cystic fibrosis is we can treat, you can squirt an aerosol of viruses containing the human protein gene into someone's lungs. It's been done in mice, and what you see is that you can correct the problem of cystic fibrosis in mice, but for about 2 weeks. And the explanation is that as a surface, an internal surface admittedly of the body, we're shedding these cells all the time from the linings of our lungs, as we do from the surface of our skin. So if you correct the gene in those cells,
I'll just go for a wee and I'll come down anyway.
And the cells you really need to transfect and correct the gene in are those in the most underlying layers, which are generating these new cells all the time, because the cells divide at the bottom, and then as they mature, they rise to the surface and then are lost. So it's a question, gene therapy will not solve all the questions if you can't get the gene you need to the cells you need it in. Okay? Right. What I wanted to do was spend a little while talking about human antibodies, because I think in medicine it would be absolutely fair to say that the most important therapeutic proteins are antibodies. Now, in the 1970s, it became feasible to make single, highly purified antibodies. And that's what monoclonal antibodies are. Have people come across that term? So it's important to realize again that although this talk is basically about genetics, there is no genetics involved in the production of a monoclonal antibody. It's confusing because it's got the word clone in, and when people hear the word clone, they think of genetics, but it's cell cloning. Nonetheless, in the 1970s, all of a sudden, the power of antibodies in the laboratory increased rapidly because single pure antibodies could be made by design. And so this is an antibody. It's one of the earlier ones, Campath-1. And that name is a big clue as to where it was made. It was made in the Department of Pathology at Cambridge University. And that's contracted into Campath, and one because it was the first one they ever made. And it was made following the interest of immunologists. So circulating around our bodies, we have all sorts of different lymphocytes with different functions in the immune system. And we have all sorts of white blood cells, some of which can be easily detected as different types of white blood cell down a microscope, but others can't be. And certainly one lymphocyte looks pretty much like another, regardless of its function. So what the immunologists were interested in is being able to distinguish different sorts of lymphocyte and monocyte in laboratory tests. Why would you need to do that? Well, in certain leukemias, you have abnormal numbers of certain types of lymphocyte and monocyte. The numbers could either drop to dangerously low level, or they could be grossly elevated because the cancer is in that particular lineage. So if you could enumerate all the different white blood cells in someone's circulatory system, you'd have a great diagnostic insight into whether or not they had leukemia and which sort of leukemia it was. So this antibody, as you see here, was raised by injecting rats originally. So it's a rat antibody. Now, as well as binding to these certain classes of lymphocyte and monocyte so that you could count them, the researchers realized that these antibodies, in the presence of suitable reagents, would actually destroy them. So instead of just using them as a diagnostic test, why not inject them into patients and use them to destroy these abnormal cells? And I hope you realize the problem with that is that if you start injecting a rat antibody into a human, after a couple of injections or so, the rat antibody is, after all, a foreign protein. The patient's immune system will start to react against it, produce antibodies. So you need to get your head around an antibody. And it will be neutralized and useless. So what you need is a human antibody with the right kind of specificity. And this is now where the genetics comes in. So we didn't need genetics to make the antibody in the first place, but what we need is the genetics to humanize it and make it suitable for therapeutic use. So here I have in black a simple structure of the rat antibody, and we can represent antibodies as these, in the simplest forms, as these Y shapes. So this is Campath1H, and it's entirely rat. Here is a human antibody. The point is that in any antibody, the critical site by which it binds strongly to its particular antigen are small regions close to the tips of these two Ys. So that's the functional bit that we need. So in Cambridge, Greg Winter in the 1980s set out to do some genetic engineering. So he's taking not the rat antibody protein, Campath1, but the genes which encode it. And then he's taking the genes of a human antibody, and by genetic engineering, he's snipping out the critical binding site regions near the ends of the arms from the rat antibody, so that gives the correct binding properties, and he's slotting them into the framework of a human antibody. So now we have a humanized antibody. Most of it is human in origin, and only the binding sites were derived from the rat. So that's all done at the DNA level by genetic engineering. So now we have Campath1H, the H being designating the fact that it's been humanized. And they took a leukemia patient, put them on a course of injections of Campath1H, and the patient was cured. And that has now become a routine approach for those particular, for leukemia of that particular sort. So the antibody started off, it was raised as an antibody, as a protein. Now it's been genetically engineered. Okay? So Winter and his colleagues could have sat back on their laurels and would be deemed very successful medical scientists, but they didn't. What they realized is that you could dispense with the rat's immune system entirely. Now, the genetics of antibodies are very complicated, and I could easily spend an hour walking you through that. The point about antibodies is that potentially we can make an almost limitless array of them. So we can do this because our antibody genes are not intact. They're an array of fragments. And the immune system is randomly combining these different fragments, and whichever antibodies are needed become selected for production. So there isn't a gene for an antibody. There are arrays of genes which together can produce any antibody the body might require during our lifetimes. So Winter realized that if he could isolate those gene arrays and put them in bacterial cultures and have all the selection and recombination of these fragments to make any antibody take place in the test tube, we can dispense with the human immune system completely. So this is entirely done by taking the DNA of these fragments, totally dependent on genetic methods. And so they produced, for example, an antibody called Humira. And this is a blockbuster drug for the treatment of rheumatoid arthritis. So rheumatoid arthritis, again, not a genetic disease. It's an inflammatory condition of the joints where the joints become progressively destroyed because of excessive inflammatory reactions taking place in them. And because we know about all the proteins of the inflammatory pathway, largely through the application of genetics in research, incidentally, Humira was developed to target a key protein in the inflammatory pathway. And it works remarkably successfully in blocking the inflammatory episodes which would otherwise be working to destroy the joints of people with rheumatoid arthritis. Now, rheumatoid arthritis is a common disease, disease of older years, but there's a huge market for a successful therapeutic agent to treat this disease. So when I say blockbuster drug, we're talking of millions of dollars of sales a year. So this is not rare, interesting medical science. This is big business, big pharma, lots of patients being successfully treated. And all because of our understanding of antibody genes and our ability to manipulate them. And it should be said that if you scan the best-selling list of therapeutic agents these days, humanized antibodies of this sort, made in this kind of way, pop up quite frequently on that list. So a major field of medicine made possible. And because of his contributions, Greg Winter was awarded the Nobel Prize for Chemistry in 2018, and also he got a knighthood. So here is another field of medicine which depends on antibodies. So this field is called cancer immunotherapy. Now, what has emerged over recent decades, we're all aware that the immune system protects us against viruses and bacteria, right? But there's been a growing awareness that perhaps in a more behind-the-scenes way, our immune system can protect us against cancer. Now, this gives us a completely different perspective on cancer, because what it means is that perhaps we're developing cancers all our lives, but our immune system is busy at work wiping them out before they become a problem. So those cancers that develop far enough that a patient has to see the doctor about them will be those that have been attacked by an onslaught from our immune system, but have survived it. So if that's the case, can we beef up the immune assault on the cancer and get the patient's own immune system to attack it in a way it hasn't managed before? And one approach to this is the field of checkpoint inhibitors. So if you're driving a car, I don't know how many have taken driving lessons yet, there's two ways to make the car go faster. One is to use the accelerator, and the other is to stop using the brakes. And this is a stop using the brakes approach. We know that our immune system is potentially very powerful. We've already touched on that this evening. And because of that, it has positive controls that fire it up, but it must have negative controls to stop it when it's inappropriate. We've already considered that type 1 diabetes is an autoimmune disease. So that's a situation where the immune system has to attack specific cells in the body. By and large, that doesn't really occur. And it doesn't occur because the negative control, the brakes on the immune system, are quite efficient. Now, we've learned a lot about the immune system, and genetic research techniques have been very important in that, incidentally. But we've come to understand what the proteins on the cells of the immune system that represent the braking system are. So let's go out and block them, and that's the equivalent of blocking the brakes. So this was a study which, as you can see, was performed in 2015. And it is on melanoma, a form of skin cancer, which those of us who have pale skins are very prone to. Excess UV light, because we lie on the beach too much or use sunbeds, can attack the melanocytes, and they can become cancerous. This isn't just a problem of the skin, because unfortunately melanoma can become metastatic, spread through the body, and is no longer operable upon. So here we see patients enrolled in this trial, and that if they're given, as in this red line, one particular antibody, and it has to be a humanized antibody because regular doses are being given. They have these very long fancy names which I struggle to pronounce, so I'll pass on that. Then we got a cure rate in these patients of about 25%. If we use two slightly different antibodies with different targets, then we get a cure rate of over 50%. Now, what you need to realize that this was an early trial. And in the early trials, the patients enrolled on it will be those who've already been through all the available conventional therapies and are essentially beyond hope to existing approaches. So even in this early trial, 60% almost of those patients were cured. Now, this timescale across the bottom doesn't go out that far, just barely a year, but the responses to this treatment have proved durable. So this is a remarkable success for an early trial. So much so that it was only 3 years later from this trial that two workers behind this approach, James Allison in America and working independently in Japan, Tezuku Honjo, were jointly awarded the Nobel Prize for Medicine in 2018. Normally, it takes many years before people decide a piece of research is worthy of a Nobel Prize. And the speed of this award is an indication of the excitement which this has generated. And already this kind of approach is in routine use for melanoma and some other cancers, and people are very hard at work trying to improve on the success rate and to try and spread it across different fields of cancer. So the underlying message is it's based on humanized antibodies. Without a considerable knowledge of the immune system and power of genetic analysis, none of this would be possible. Okay? So let's leave medicine behind and go on to DNA fingerprinting. So it's very difficult to think of a serious criminal investigation that wouldn't try and use DNA fingerprinting as a technique of investigation. Now I think it was about November on ITV, there was a 2-part drama based on this story. I don't know if people saw it, it's probably still available on catch-up. But the story starts with this fellow, who was then a young lecturer in Leicester, Alec Jeffreys. And it's interesting that despite its enormous application to criminal justice, Jeffreys was interested in science. And he was a geneticist interested in the genetic variation between individuals. What differences are there between populations of the same species? And we've already touched on the fact that some people are unfortunate enough to inherit a mutant factor VIII gene and therefore have haemophilia. Most of us have a completely normal gene. So there is a certain level of genetic variation across the population. Not all of it results in disease in that way. Some of it's just normal variation. But if you look in the genes which encode proteins, there isn't that much variation. But if you look outside the coding regions into regulatory regions of the genome, you find huge variation. So I don't know if you've come across it, but there are regions between the genes called mini-satellites. And there are about 1,000 of these in our genomes. So they have some kind of regulatory role in gene expression that we don't fully understand. But one thing that Jeffreys realized is that there's considerable variation between these between one individual and another, not just in sequence, but actually in length. These are highly repetitive regions, and even the number of repeats that we inherit from our parents differ. And of course, because we have 2 sets, one from each of our parents, each of us as individuals has a really unique makeup of our mini-satellites. So we can time his discovery very precisely at just past 9 o'clock on Monday, the 10th of September, 1984. And the reason for that exact timing is he'd left an experiment to develop over the weekend. And first thing on the Monday morning, he went in, developed his experiment, and bingo. And here he is showing, proudly as he might, the results of such an experiment. So what's happened is that a group of different individuals have donated blood from which DNA has been extracted. The mini-satellite regions have been cut out and amplified up many times so they can be detected. And they're run out on a size separation. So he's holding up the results. Each vertical track here, I think there's 13 of them, is the sample from a different individual. And basically, the big pieces don't get very far from the top downwards, so they stay up the top. The average size ones are in the middle here, and the little repeats will have run a long way down to the bottom. So it's a size separation from top to bottom. And without any sophisticated understanding, all you can do is eyeball those results, and you see that none of those 13 vertical tracks are exactly the same. Some of them are quite similar. Some of these spots turn up in virtually all of the tracks, but essentially the combination pattern of these bands is different in every single individual. So Jeffries then realized he had a way of uniquely identifying every individual in the population. Now, the story that's told in that TV dramatization is rather a grim one. In 1985, a young teenager was raped and murdered in the village of Narborough, which is just outside Leicester, where Jeffries worked. And more tragically still, a few years earlier, a very similar crime had occurred, another rape and murder, in much the same area. So the fear was that there was a serial rapist and killer on the loose. And the police ran out of clues to this. They called in Jeffries, and he was able to show from samples of both crime scenes that indeed the same individual was involved in both crimes. So the worst fears of the community were realized. He also showed that the microsatellite pattern of the police's chief suspect failed to match. So that particular man was innocent. But that left the police with no suspect and no clues. Nonetheless, they were so impressed with his results that they organized what was the first DNA manhunt ever to take place in the world. They asked the men in the neighborhood to give samples of DNA for analysis. And because the community was so traumatized, they were only too willing to comply. But the results of screening thousands of samples failed to find a match. But then one night in a pub, someone was overheard saying that he'd given a DNA sample on behalf of a work colleague. And that conversation was reported to the police. The person in question was rounded up and indeed found to have the perfect match for the DNA. So that conviction, and indeed the identification of the suspect in the first place, was entirely down to this fingerprinting method. So at that stage, police and crime authorities worldwide took notice, and most countries began collecting DNA from criminal suspects and building up national database, national databases of DNA patterns, so that in future crimes they would have a resource with which to match the DNA found at crime scenes. There are ethical issues associated with that, and most countries, such as our own, have now legislated as to how far that should be allowed to go. So I think it's getting quite late in the day, but I did say I'd touch on emerging viruses. So just to summarize this particular slide very quickly, we have seen in our rather overpopulated and highly mobile world a number of outbreaks of new viruses emerging. But of course, what our current preoccupation is with SARS-CoV-2, which has already caused 5.5 million deaths and rising. So, can genetics help us here? And the answer, very obviously, is absolutely it can. So the mere fact that this was a new virus as opposed to a re-emergence of an old one was only identified when, and ambiguously, when the genome of the virus was sequenced. And that took place very early on in the outbreak of this pandemic. So we knew we had a new viral threat. Since then, of course, I'm sure, again, you're all highly familiar with the fact that we have had waves of the epidemic because of new mutant strains of the virus. How can we confirm that this is really a new mutant strain? And the answer is genomic sequencing, which of course can be done virtually overnight with a fully automated sequencing machine. Once we know about that, we can use genome sequencing to monitor the spread of the pandemic around the world and into communities. And of course, you're all highly familiar with the idea of polymerized chain reactions, PCRs. I won't go into the details, but essentially it's a genetic technique for amplifying up sequences of interest. And even our lateral flow tests that we're all taking, they require antibodies in the test kits. How can you make those antibodies? You isolate the genes of the virus, put them into culture methods. From that, you isolate large quantities of viral protein, and you can make your lateral flow tests. And then moving along, to fight the virus, we now have genetic vaccines. So it's perfectly possible with COVID-2 to make conventional vaccines. You develop a large culture of the virus, isolate the virus and its proteins, then inactivate it and use that as your virus. This is a standard vaccine manufacturing approach that's been used for decades. Not really any genetics involved in it, and it does work. It's an approach the Russians and the Chinese have taken with their vaccines. But we're fortunate that we have these genetic vaccines which now appear to be working much better and to provide much better protection. So both BioNTech and Pfizer in their collaboration, and also Moderna, have generated a new type of genetic vaccine based on RNA. So the RNA encodes the spike protein of the virus. You manufacture that into a virus-like particle. It isn't a virus, it cannot cause an infection. But when injected into the body, it will enter cells like a virus would do. And the RNA persists long enough to for the cells to make the viral protein themselves. That's recognized by our immune systems, and we get protection. The Oxford AstraZeneca virus takes a slightly different route and a slightly more established one, and that's using DNA. So it's still the same gene, but for the spike protein of the virus, but in DNA form. That DNA is inserted into an adenovirus. That's a fairly harmless virus. And in the case of the Oxford vaccine, it's particularly harmless in humans because it's a chimpanzee virus. It causes only a very transient infection, so it's soon cleared, but it will leave our immune system with a memory of the spike protein, which was encoded within it in this piece of DNA. So these highly successful viruses, which are what is now allowing us to resume something like normal activity, have all depended on sophisticated genetic research. Okay, so thank you for your attention. I'll finish there. So I'm happy to take questions on what we've considered. Yeah.
One of the slides you discussed about how slowing down the immune system.
Yes.
In the cancer, but wouldn't that be a bad thing because that's going to enable harm neighboring cells?
Right. Right. So the question is taking the brakes off the immune system harmful?
Yes.
The answer is yes, it is. It's not a sort of no pain answer. It's not so much the neighboring cells because the immune system is pretty well focused on the abnormal cells. But what it does mean is that patients can suffer from severe flu-like conditions because you can invoke a very powerful immune response at the moment. I mean, perhaps we can get more sophisticated about this in the future and do things on a slower time scale, but still eliminate the cancer. It is not without danger, and so it does need to be progressed with caution. So there was another question. Okay, right, must be a good one then.
I think it was back in 2018, there was a researcher in China who announced that
Yes.
make them immune to HIV, and then there was a lot of outrage from
Yes.
the community. Why was that?
So the question is about a report that a Chinese man had genetically engineered twins to make them resistant to HIV. So I think there's several answers to why this caused outrage. And in fact, the guy was arrested and sentenced and imprisoned. It's not actually clear to me that he actually did it. He claims to have done it, but there's been no publication of his results at all. It certainly wasn't for a long period of time. But one of the real issues is that he claims to have engineered them to make them resistant to HIV. So the point is, you can make yourself resistant to HIV by acting with caution in your sexual contacts. So why do you need to genetically engineer someone to have that same effect? And on the downside, can we absolutely, this is a normal gene which he is sticking a mutation in. So can we be 100% certain that there is not some consequence of this tampering with the gene, which is actually an important one in the immune system? And the jury is out on that. There have been some studies that say this mutation, which does occur naturally, incidentally, and that's why some people have proved resistant to HIV infection. The question is, do they have other consequences of that mutation? I think the jury's out on that. So to give someone deliberately a lifetime of absence of that protein, it's a bit risky. Okay.
Through the chat box, it's a bit more difficult to communicate, but we can ask questions still through the Padlet on the PowerPoint presentation. And thank you so much, Chris.
A pleasure. I hope you found some of it interesting, at least.